Assalamu alaikum warahmatullah. In a series of lectures concerning the course Applied Pharmacognosy, the course designed for 5th year pharmacy student. In our first lecture of this course, we will talk about methods for analysis of medicinal plant, mainly the use of macroscopical, microscopical, and histochemical tests for assessing medicinal plant. This lecture deals with the use of various physical and chemical methods to assess or to determine the identity of medicinal plant and determine the presence of important class of secondary metabolites which are important for their therapeutic uses. So the main of this lecture is to express or describe the method used for two purposes. One of them determining the identity or the identity of the medicinal plant used for therapeutic uses, determining the presence of important classes of secondary metabolites. As we know, we mean by secondary metabolite is important class of compound that occur in plant and biosynthesized in plant and other living cells, but they are not essential for growth for the plant, but have other function. They are used as a defense mechanism or as attractant for predators. For humans, these secondary metabolites having pharmacological activities or therapeutic value. The lecture consists of three parts: the use of macroscopical test, and second part, the use of microscopical test, and the third one, use of histochemical examination. We mean by macroscopical examination using of sensory organs اللي هي أعضاء الحواس to determine the identity of secondary of the medicinal plant itself. باستخدام الشم، اللمس، اللون، الرائحة عشان نحدد هوية النبات. In microscopical examination, we mean by using microscopy to determine the infrastructure of the medicinal plant in order to determine its identity and besides to determine the presence of secondary metabolite in this anatomical structure. And the third part of the lecture, we talk about histochemical examination, and we mean the use of microscopy beside chemical reaction to identify the identity of type of secondary metabolite that occur in this medicinal plant. In details, we talk about 12 classes of secondary metabolite, or in histochemical examination, we study the use of chemical reaction and microscopy to determine the identity of 12 important classes of secondary metabolites. In the first part of this lecture, we will talk about macroscopical analysis of medicinal plants. As the name implies, macroscopical analysis means the use of visual inspection to determine the identity of medicinal plant, either by naked eye or using magnifying glass. In fact, macroscopical analysis used for analyzing organized drugs that can be noted or can be showed by naked eye, like the organized drugs such as drug from roots, drug from bark, drug from seeds, flowers, and so on. So the examination helps in identifying the plant material using their morphological or organoleptic characters اللي هي الخصائص الظاهرية أو الخصائص عندها علاقة أو ممكن معرفتها بالحواس، الرائحة، اللون، الملمس and so on. So, macroscopical analysis procedure for medicinal plant material can be divided into two subclasses. Method for analyzing or for analysis of organized drugs, such as drug from roots, drug from seeds, drug from flower, and so on. And the other part for unorganized drugs, such as gum and residues, besides the powdered drugs. For organized drugs, the procedure for analysis based on the first step is sample selection, which means collect representative sample of plant material that may be roots, stem, flower, leaves, and so on. So the next step is observation or preliminary test, which means examining sample under good light condition to observe the following characters. And here the shape, size, color, texture, surface features, if there is ridge, veins, or hairs, using or noting also other tests and noting the fracture, the behavior when this part is broken, is it brittle or fibrous. And we talk about the fracture behavior in details when we talk about drug from bark and drug from wood. So the next methods for unorganized, we directly observe, starting from number 2, observation. We take sample, we good light condition to observe the following characters: the color, other, the taste, and if it is powder mixed with water, or if it is mucilage, to note mixed with water to note the presence of mucilage, or mixed with sulfuric acid to know the presence, or to show the presence of chalk, and addition of organic solvent to show the solubility of unorganized drugs. So, at the end of this slide, we means macroscopic analysis can be carried by two ways or by two methods. For organized drugs, select sample which to be analyzed or identified, is it leaf or root or stem, and so on, and then we know their characters in good lightening condition to show the color, size, shape, texture, and so on. For unorganized, we directly observe the sample under a good light condition, and we know the color, other taste, beside other specific tests for powder organized drugs, the use of water, the use of sulfuric acid, and use of organic solvent. And note the difference in observations to be identified for organized and for unorganized drug. For macroscopical examination for organized drugs, there are different characters or features depending on the part of the drug used or the plant used. We can do observation or macroscopical examination. As we know, there are difference in feature between leaves, flower, seeds, and fruits. By naked eye or by using a magnifying lens on the leaf, we can show the following characters. Besides the color and shape is the same, color, shape, and taste is the same for the four organs or four parts, but for the shape and for the leaves, we have a specific feature on the shape. We can show, or we can examine the type of margin. Is it entire? Is it serrate? Is it dead? And so on. and the apex of the leaf also is important for differentiation or examination. The type of venation نوع التعلق هل هو reticulate هل هو بالنت أو بالنت parallel and so on. Also the base of the leaf and the side surface, texture, color, other and taste. For flower, as we know, the composition or the part involved in forming flower is different from that of the leaves. The first feature is the type of inflorescence for flower. نوع النورة هل هي راسيموز أو سايموز هل محدودة ولا غير محدودة. التايب of flower itself هل هي zygomorphic هل هي متناظرة أو غير متناظرة and so on. الكاليس also is important for characters corolla and gynosium and androsium and gynosium also is important. As we know, الأندروسيوم هو consists of عضو التسكير اللي هو بين الملك والقلم الأنثر عن فيلامنت. الجينوسيوم عضو المؤنث متكون من الأوفري والستيغما والستايل. كذلك الأوفري within ovary is important for identification of flower. For the seeds, the important features we note the funicle or المشيمة. We know the hilum and microbile, and we know the difference between hilum and microbile. Hilum is, or both of them are scarce on the surface of the testa, while hilum is a spot for the joining between the seed and funiculus اللي هي داخل الثمرة ارتباط between البذرة والثمرة من خلال المشيمة. This is the hilum, was scarlet by removal of funiculus, while microbile is a small pore in the testa surface that through which the pollen grain enter into the ovule for fertilization. Also the seed coat, number of seed coats determine the type of seed. Pre-sperm and endosperm, also this at the part below the seed coat, and the embryo. Also the shape of embryo can determine the type of the seed, if it is folded or it is straight and so on. For fruit, we determine the type or class of fruit. We have different type of fruit, true fruit and false fruit, a shape and dimension, adhesion, adhesons. This for dry fruit زي ما في الأمبريفي fruit الكورياندر والفانيل والأنايس هما adhesant can be separated into two parts يسموهم الميريكارب. البريكارب also اللي هو الغشاء المحيط للثمرة مهم في الidentification. Placentation also again the seed, all the features of seed can be described in fruits beside the other and taste. The preliminary test for powdered drugs and unorganized drugs also. The important features or characters we can observe in the powder or the bulk of the material. We note the color. The source of the blood determines the color of the powdered drug. Here below there are several examples for natural drugs powdered form with different colors. For example, cassia and calcium, they have white colors, while الكوليسانس والليكورايز في الجنجر كواسي and still having and squid having yellow color and other brown color زي ما قلنا until licorice نص vomica opium powder vanil Lucian and so on. We can go through this different type of powder drugs. We know their color. We know that they are in the powder form. This color for powder drugs in powder form. Also, another important preliminary test for powder drugs is their other observation or not their others. There is some example for powder drugs having characteristic other. We know by going to the reference material or the reference for these natural drugs. Also, another third important morphological character can be noted for powder drugs is their taste. But it's important to be noticed that the adulterated sample may be harmful, contain toxic drugs, or may be too pungent to be tested, such as capsicum, التشيلي أو الشطة هي لاذعة جدا، ممكن تكون مؤذية بالنسبة لنا when tested أو نعمل شنو عندما نتذوق. Also can be too toxic like drug containing alcohol. So the student should not test about that drug without the consent of their supervisors. When in the lab, you are not allowed to test any powder sample unless you consult your supervisor or the demonstrator. In general, there are some examples for powder drugs having aromatic taste, such as coriander, cardamom, cinnamon, clove, and nutmeg, and other with aromatic taste and also aromatic and pungent taste, such as ginger and capsicum. We have another preliminary test for powdered drugs and unorganized drugs because they are maybe toxic or too pungent or harmful to be tested. We have another means to detect the identity of the powder sample. One of them is mixing the powder with few drops of water and allow to stand. And for this test, we know or we note the nature of the powder. If it contains gums, the appearance become more homogeneous or viscous in nature. And if we mix the blood sample with sulfuric acid, we note what happened to the sample if البيزلز في فوران معناتها السامبل containing chalk or containing calcium carbonate. Also, for powdered and unorganized drugs, we test the presence of fixed oil and volatile oil in the powder sample by pressing the powder between two filter papers. We bring two filter paper and we place the powder, small quantity of powder between the two papers, and we apply light press, نعمل ضغط خفيف, and we note the oil stain. If it is present oil stain and resistant for long period of time, and when heated in oven, that means the stain or the sample is containing fixed oil. If the oil is volatile, then the stain disappears on heating in oven. بالنسبة للpresence of fixed oil and volatile oil. We press the powder between two filter papers and we note the spot. If it's resistant on heating, it is fixed oil. And if it is disappear or evaporated, that means it is volatile oil. The last test for powder sample, the preliminary test, we shake the powder with half test tube with water from the water sample, and we shake the sample and note the frothing occur. The presence of froth or formation of foam and the this means the sample containing sabonine compound. And when we boil gently, note the other of volatile oil evolve. So we have another test for to confirm the presence of volatile oil besides the use of filter paper is by shaking the sample with water and heating or boiling, and to note the evolution or the evaporation of the oil, and we note it by its smell. That means the sample contains volatile oil. But if it is formed froth, that means the sample containing sabon. The second part of this lecture is the use of microscopical techniques to identify or analyze crude drugs or missile drugs. As the name implies, microscopical analysis involves the use of microscope in order to identify the microstructure of these medicinal plants. This examination helps identifying the plant material that deposited or related in these cells or these tissues. So microscopy can be used for determining the microstructure of plant and identifying the presence of plant material or secondary metabolite within the tissue of the plant, or this can be used to determine the identity of the used plant. For microscopical examination, there are several sequential steps or procedures can be followed to determine the cellular content and the microstructure of plant sample. First of all is sample collection. That means collecting the representative sample of plant part to be examined. If it is the leaf or flower, stem or seed, and so on. The second step is fixation, and this can be carried out by using fixative solution such as formaldehyde to preserve their cellular structure. The second step is sectioning. This means forming sections, or if it is a transverse section or a section, using suitable sectioning equipment depending on its nature. If it is, we use microtome or razor blade. Razor blade can be used for soft tissues like leaves or flower, while microtome can be used to make section of hard tissues such as bark or wood. After sectioning, the fourth step is staining by using appropriate chemical reagents or dyes to highlight specific structures. The last step before observing under microscope is mounting. Mounting means use mounting agent to fix the sample on slides, and after that we add cover slip to be visualized under the microscope. Example of mounting agent used is glycerin. So the importance or the purpose of adding mounting agent is to aid visualization of the sample, and the microscope also can provide some type of preservation for cutting or prepared sample to be visualized. These are the type of sectioning tools can be used to prepare the section of the sample, either using razor for soft tissues after moistening or after boiling the sample. Soft tissue like leaves, roots, and bark, young bark or young roots after boiling can be cut by razor, or by using freeze microtome, also can be used to prepare botanical section, and it's especially for hard tissue like older bark and stems or wood. In order to visualize or examine the distribution of tissue in the plant sample, we can make a section. There are different types of sectioning and different type of section obtained, either transverse section or L section, based on the way of cutting. Always the transverse section making section as is in this picture. We make a section at the middle or at the radius of the stem or the plant sample, or either we use a vertical line to obtain an section, as appear in these pictures. After preparation of the L section and T section, the sample or the section should be mounted on water or dilute glycerin to preserve its structure and to aid its visualization. Subsequently, this section can be cleared by using clearing agents such as chlorohydrate to remove the stored material such as carbohydrate and fish oil to aid the visualization of tissue distribution under microscope. Also, disintegration agents such as formic acid can be added to section to aid the isolation of specific tissues, especially the tracheids and conducting vessels. Bleaching agent can be added to remove colored material, and defatting agents or techniques can be used to remove fats from the seeds. In general, clearing agents can be used together with different staining agents to stain the cell wall and the cell content. In practice, we use clearing agent to remove fats, and we add staining agent to stain the remaining tissues such as cell wall and cell content can be aided by using staining agent. But the bleaching agent and disintegrating agent can be added in some cases if the nature of tissue is required this type of reagent. There is some example for microscopical reagent that are used for examination of D, S, and L section of plant material. Ethanol can be used as preservative, also can be used as clearing agent similar to chlorohydrate to remove oils and resins. وعنده ميزة إنه cannot dissolve gums and mucilage. So it is a suitable agent if you need to visualize gums and mucilage to add ethanol because it removes other material and left gums and mucilage as it is. Alkana root, it is staining agent, can be useful for fats, oil, and superized and cuticularized walls. Chlorohydrate with glycerin is useful clearing agent also. Besides the presence of glycerin with chlorohydrate can aid the preservation or retention of chlorohydrate for several days without evaporation. يعني لو عايزين نعمل clearing في نفس الوقت عايزين نحافظ على structure of our slide أو our sample, we can use chlorohydrate and glycerin. والميزة الثانية إنه بيعمل clearing من غير ما نحتاج لإنه للتسخين. دائما الكلوروهيدرات عشان نعمل bleaching أو clearing يتطلب استخدام الحرارة أو التسخين. Carbohydrate is known as clearing agent. The chromic acid also it is microscopical reagents can be used for disintegration and isolation of tissues such as isolation of البلدز والتراكايد والconductive tissue in general. Also chromic acid and nitric acid mixture can be used as disintegrating agents. Clove oil used as clearing agent. Similarly, carbohydrate ethanol, but clove useful for examining plant sample containing oil. Ethanol useful for mucilage, while clove oil used for sample containing oil. Cresol is a macroscopical reagent suitable for mounting sample of chalk and kettle for powder sample or for minerals containing material. Chlorozinc iodine, also known as Sewell's solution, وهو مناسب ل identification of cellulose in the cell wall. Also, the peroxide ammoniac solution, known as coxam. Its importance is cause swelling and formation of solution لcellulose wall. تعمل لها swelling وcan form solution أو بنسميها dissolving, can be used for visualizing أو examining the cell wall cellulose, and بنسميه swelling بنسميه تضخم, can be observed clearly. One of the problems with coxam is after a period of time or specified time can dissolve the cellulose, and it should be used with caution, not to be allowed to stand for long time without use. The reagent coralline and alkaline solution, known as coralline soda, is thin reagent for cellulose of sieve tubes, also suitable for some type of gums and mucilage. Ether is another reagent, is useful as defatting agent, can be used as clearing agent to remove fat from the sample. Sudan Syri or Sudan Red were useful reagent to stain or sample for presence of oil تحديدا fixed oil and essential oil mainly والاثنين الاسينشال اويل في الستورج تيشوز اللي هي في السوبرايز وولز او في السكرتري cells and secretory duct. As we know, essential oil can be stored in different anatomical structures. ممكن يكون في الوول of الwood gland زي ما في الساندل وود أو كان في secretory cells أو secretory ducts زي ما في الجنجل والكلوف أو إيفن في ترايكوم زي ما في أوراق النعناع اللي هو المنتا بايبريتا أو البيبرمنت leaves. The last microscopic reagent البوتاسيوم تترا أيودو mercurate اللي هو known as Mayer reagents وده well-known reagents using for staining blood-containing alcohols. Histochemical examination of cell content means the use of chemical reagent to examine the histochemical structure or the microstructure of the blood sample. Always histochemical examination followed preparation of sample as in microscopical examination, formation of the trabecular section or section. Also, histochemical examination is applicable for cutted sample, dissection or resection, for powdered material or for organized. It is involved or it is carried out by applying drop of the reagent on the sample. Then we add covered glass and examine under the microscope. In this slide, we summarize the histochemical test for 13 important classes of secondary metabolites that present in plant sample. It started with cellulose and end with tannins. Cellulose can be stained by using two tests, either by iodine zinc chloride or sulfuric acid test, وده بيدينا blue to white stain، أو باستخدام الكوكزام test، والكوكزام give أو بيحصل swelling للcell wall containing cellulose and gradually dissolve when the time is going on. Lignified cell walls that contain lignin can be stained by using fluorogesol reagent and HCl, and with fluorogesol lignified cell wall give pink to cherry red color. Superized and critical containing cell wall can be tested by Sudan 3 reagent. They give red or orange red color. Alluron grains or protein storage vacuums that contain protein can be tested by 3 different tests, either by iodine ethanol or ethanol trinitrophenol or mercuric nitrate. Alluron grains with iodine and trinitrophenol reagent give yellow color, while for iodine give yellowish brown to brown color, and for trinitrophenol give yellow color, while with mercuric iodide give brick red color. Calcium carbonate, its presence can be detected by using acetic acid and HCl solution. When we add this reagent to sample containing calcium carbonate, then the calcium carbonate will dissolve slowly and forming effervescence. While calcium oxalate can be tested by 3 different reagents, either by acetic acid or HCl or sulfuric acid. Calcium oxalate in acetic acid are insoluble, and when we add HCl, they dissolve with formation of effervescence, and with sulfuric acid, they form a shaped crystal, calcium sulfate, separate on standing after about 10 minutes. Again, fatty acids and volatile oil acids can be stained or identified by Sudan 3 ethanol reagent, and both of them give orange red to red color. Fixed oil and fatty acid and resins, all of them give red color. While with fatty acid, the color remains intact when we add ethanol, but it dissolves in addition of ethanol if the sample is containing volatile oil or resins. Because ethanol dissolves volatile oil but not dissolving fixed oil. Hydroxyalpha-quinone, these are natural dyes, and some of them have pharmacological activities such as sinusoid and imodine and phythione. All these are anthraquinone compounds having pharmacological activities, most of them used as laxative. Hydroxyalpha-quinone can be detected by addition of potassium hydroxide. They give red color with potassium hydroxide. Inulin is a polysaccharide, can be detected by using alpha-napsol and sulfuric acid reagent. Inulin, with addition of napsol and sulfuric acid, form a spherical aggregation of crystals of inulin, can be changed to brownish red color, and lastly they are dissolved when time is going. Mucilage and gums can be tested by Chinese ink or by thionine ethanol solution. With Chinese ink, mucilage form transparent, spherical dilated fragment on black background. يتكون transparent fragment شكلها spherical with black background in case of Chinese ink. And with thionine ethanol, mucilage can be stained with violet to red color. Starch, which is different than inulin, starch can be stained with iodine, give blue or reddish blue color, and tannins as last class of secondary metabolite. It is a phenolic compound, can be stained with ferric chloride, and they give bluish black or greenish black stain. By the end of this lecture, we know the different methods, physical and chemical methods used for analyzing plant material. Actually, we speak about macroscopical examination, microscopy, and histochemical examination. The main purpose of this, of the aim of these reactions, of this test, is to determine the content of the secondary metabolite content of plant sample, also to determine the microstructure of plant sample, and at the end we determine the identity of the plant sample. Macroscopy will depend on the studying the sensory characters by using sensory organs, study organic properties such as color, odor, and taste. In microscopy, we use a microscope to study the microstructure of plant sample, and also to examine the presence of secondary metabolite in these microstructures. Histochemical examination it is complementary to microscopy examination. We use chemical reagent to stain these tissues by using dyes or by creation of the color change of the transverse section or L section.